Hello. Today I'm going to talk about another characteristic that highlights senescence-inducing factors. In a previous article, I talked about proteins that live on the basolateral part of the membrane of cells. Those I call the proteins of aging. And in fact, when you have factors like protein or other small molecules that bind to the basolateral membrane and induce aging, they bind to those proteins that live there. What are senescence-inducing factors? Those are factors like interleukin-6, tumor necrosis factor. Those factors, when injected into the plasma of experimental animals, induce aging in the animal, or in a experimental lab situation, they induce senescence in cells. The interesting thing about senescence-inducing factors is that many of them have a very low isoelectric point. What's the isoelectric point? That's the point at which, when that pH is there, the acid and base numbers in the protein are equal. For example, an isoelectric point of 7 means that at pH of 7, there's an equal number of acidic and basic residues on the protein. Many of the senescence-inducing factors have lower than 7 isoelectric points. One of the really problematic senescence-inducing factors is lipopolysaccharide. This is part of a bacterial cell wall, a gram-negative bacteria, and induces havoc within the organism when the host undergoes gram-negative sepsis. Because of this lipopolysaccharide, the whole system goes awry. The isoelectric point of lipopolysaccharide is 2.5. It's extremely low. I put several examples of proteins that have low isoelectric points and that induce senescence in cells. Interestingly, each of those proteins or factors bind to receptors on the basolateral part of the cell. Thus, here we have two characteristics of senescence-inducing systems. One is the senescence-inducing factor that tends to have a low isoelectric point, and the second, those senescence-inducing factors bind to proteins that tend to localize on the basolateral membranes.